Welcome to the Australian Human Microbiome Biobank. When we receive your sample, it is first de-identified to remove any connection to you, the donor. We give each sample a unique code and transfer it into the AHMB lab for accessioning, where we sort the samples and record important details about them, and enter it into our lab software.
In order to keep track of our sample information, we use a Lab Inventory Management System, or LIMS. We have built a custom LIMS to maintain sample tracking through our whole workflow and to ensure strict quality control of samples and isolates. We can see the sample's unique code. It is now officially part of the AHMB and can move into our first processing step.
A critical step in our biobank's ability to isolate novel and interesting microbes is our capacity to separate individual microbial cells, or isolates. Your sample arrives as a mixture of microbes and debris. This flow sorter allows us to separate them out to grow them individually. We are able to use information gathered from 5 different lasers to select microbes of interest and deposit 96 individual microbes onto a plate.
Plates of individual microbes are then transferred into our custom-built anaerobic chamber. Since the majority of microbes from the human microbiome are oxygen sensitive, this chamber allows us to control the atmosphere we grow them in. After a few days, the single microbes will grow into small colonies.
When the small colonies have grown on the plate, we move them onto the Pixel, our automated microbial colony picker. This ultra-reliable robot facilitates our high-throughput process and allows us to identify microbial colonies to easily transfer them to the next steps. Now, they incubate and grow for up to 3 weeks.
Once the isolates have grown, they are ready for banking. We transfer them to our 2D barcoded tubes, which allow for each isolate to be reliably identified and tracked through the lab. The information from these tubes gets added to our LIMS, so now we have each unique isolate in a uniquely coded tube.
The final step is adding our newly made isolates to our inventory. We have ultra-low temperature freezers which allow us to store microbes long-term at -80 degrees Celsius.
This process is allowing our team to build the world's first high-throughput cultivation platform for biobanking human microbes. Find out more at ahmb.com.au.